Yeah, so today we are at week 11 of the course for the session for Introduction to Proteogenomics. So we have looked upon many things starting from the week one, like the different genomics technologies and proteomics technologies, and now we are mostly looking into how these things can be integrated. So here today we would be looking at the very first, very crucial aspect of the any kind of proteomics or the transcriptomics data analysis is that to look at the identify the pathways associated with those particular diseases or the perturbation of those particular things. Secondly, that thing that which we are would be mostly looking into it, that the in the genomics technologies we have looked at SNPs or the single nucleotide polymorphism, but how it affects when it comes at the proteome level or specifically at those kinases where the mutations happen. So how we can get it. Now the first question is to understand if we are doing this kind of big data analysis or the omics data analysis where we are looking at the entire genome or the proteome or transcriptome of an organism. So definitely the first thing that why we have started looking at each and every full genome and proteome comes from the fact that the no your not a single gene is able to work alone or and is surrounded by a series of networks. So if one second. Yeah, hopefully it's yeah, hopefully it's visible now. So now we are doing is that looking at the different aspects. So the concept of full genome or the proteome is to understand that how all the things work in unison, that while perturbing one of a particular gene or protein, how it is going to affect other regions or other proteins and how it is affecting as a whole. So that's why the concept is coming of the network to understand the network. So that's why once we have done with the analysis, we need to understand that how entire network is getting disturbed or perturbed. So that's why the concept came like the for no single gene is can work alone or how it can take it forward. So the network based approach to human disease. So this concept came that the genes that are associated with a particular disease, so there will be some proteins or some genes that are directly linked to for a disease condition, and if that disease and that sets gets perturbed or gets differentially regulated, so that will have a huge impact on the human body. So here, among the 25,000 genes of the human body, so there are around, according to their definition, they have provided that the essential genes is about 1665, essential non-disease gene is 1267, essential and the disease genes is 398, and the disease genes are 1777, whereas non-essential disease genes are 1379. So these are the some kind of fabrication or segregation of the different kinds of genes that could have been done with the all the known literature and the genes that are known for causing those diseases. So you can see that the essential proteins and the disease proteins, how they plug together or they form the network. So that becomes your crucial. So here you can go to this particular uh, article and can have a look at it. So now you need to understand that how we will be forming those network or the understand that how it is affecting each other. So it can either be a topological module or functional module, or the disease module. So topologically close. So the concept here, what is the meaning of this? Uh, is that which genes are having topologically having similar functions will be associated with each other in a close cluster. So here, so the different, so when they've started doing for the different analysis and trying to cluster the different genes, so now the question definitely came that how to do this. So that's why these three uh, things came into picture. So one is there they are telling is that topologically closed genes. So the genes which are topologically closer will you will cluster each other. So definitely if you think of it, whenever the species or whenever the speciation or these variants are looked upon, so this always, so whenever, so we have discussed in detail regarding the clustering and all those things. So the definitely that which are behave same type of thing, you would try to cluster those together. Next came, this is the topological module. So afterwards you can decide on that which module works best and which module to take it forward. Next is the functionally similar genes or products. So which genes produce functional similar genes. So they can be clustered together and can be helped in forming the network. So so this was a kind of your, you can tell that it was a basis of the understanding of how to perform the networks before, if you can understand as of now, if you feed into your data and try to find out the cluster. So these are the all the basis that on which the your algorithm would work on to find out those kind of networks. So here what you will do is that you will have your functionally similar genes or products and you will place it together. Third thing was the disease genes or products, where you will be providing that your diseased genes which are causing your genes in the disease, certain disease, and you will place them together. Now in this network analysis, or these things are very much derived from your uh, graph theory, where you will have your nodes, edges, branches, and as well as the directional. So direction, so these are all, if you notice here, so if you consider it as your graph with nodes and edges and branches, so you will see that there is a uh, direction associated with it. So all these are directional, because if you go that mostly in our body, you must understand that it works in a unidirectional or sometimes in a bidirectional way as well, but mostly in a unidirectional way, where a particular gene is affecting another gene at the downstream process and meanwhile gets affected. So this is the schema of things which was taken into account when the first kind of different kinds of network, how the cluster, how the networks can be formed, and how the algorithms can be chosen forward were taken. So first one was your topologically closed genes. Second one was functionally similar genes. Third one is disease genes. So now this is like the interactome reconstruction. So potential sources that were taken was OMIM. So if you know that Mendelian inheritance genes, GWA study literature. So where you will be understanding or knowing that which particular genes are causing the disease causing, and you will place it into a particular group. Whereas if you don't take it, so from where you will be getting. So here you will see that the literature GWA study OMIM will help you to find out. Second is the third is the disease module identification. So disease one protein, disease two protein, and the overlapping protein. So if for the disease based modules, how you can uh, do that. Then is the pathway identification, known disease to protein and your predicted disease to protein. Finally, you will have your validation or prediction by using functional homogeneity, like your uh, gene ontology, tissue specificity, phenotypic similar, and your dynamic homogeneity, like co-expression, genetic interactions. So then is another thing is the disease linkage interval. where the gene 1 and the gene 2, how much is their linkage connection. So from there you determine that whether PPI will be there or not. So you can see that known disease associated protein or candidate protein or not a candidate protein. Next is the concept of the diffusion based methods. So we will be looking into it, uh, the different diffusion based methods, how the network analysis is being done through it. So known disease associated protein and the likely protein and unlikely. So you will be, so we will be looking into it in much detailed way. Yes, so this is one kind of an example of your human disease network. So here you can see that as so in a certain state of study. So if you have performed your analysis and for any kind of manuscript, if you try to look into it, so you will find out that there are many manuscripts who have definitely provided such experiments, such results in their things, where you will get a different sets of networks associated with your particular proteins or the genes which are being uh, getting away. So here you can see the networks. So this example of the overlapping proteins and overlapping genes with different other cancers. So this is for the full human disease network. So this manuscript had tried to show you the network for considering the genes and the proteins for all the diseases what was taken. So here you can see that there was around 60 diseases taken. So how that network, so you can see that most of the disease, most of the genes or the proteins will show an overlap between each other with certain genes or proteins in the downstream which is getting affected and finally leading to a particular disease. So that's why you can see that this is the network with if you do a cluster kind of analysis, you can find out that you can definitely try to find out some of those clusters of the genes which are associated for a particular disease and some of those overlapping genes like these ones, and then you can try to find their function and the correlation with that particular disease. So this was for the human disease network. Along with that, you can do a metabolic disease network. So you can have the different metabolic pathways like your glycolysis, and pentose phosphate pathway, etc., fatty acid synthesis, where you can perform according to the disease which pathways are getting affected, and then you can perform a cluster kind of analysis. So what is biological networks made up of? So whatever we talk about, and if it so ultimately whenever you do a for example a proteogenomics based study, along with the identification of the variants or the different therapeutic candidates, can be immunogenic candidates or other things. Also we need to understand the different pathways and the things that are being shown uniquely by that particular patient or a particular disease. So whenever, so these things what we were looking at, this all these different complex graphs and those things, so actually what it is made up of. So this is actually a simpler representation of what is there. So in the biology we know that we have a protein, we have a gene, we have a metabolite. So we will have a PPI, that is your protein-protein interaction. So protein-protein interaction would be playing a huge role. Second is the genetic interaction, DNA-protein interactions, gene regulation, cell signaling, and cell metabolism. So a biological network consists of all these parts, which is represented in the form of graphs in whatever you can see. So whenever you go through any manuscript kind of thing, so you will find out that there are many networks and those tools that are present here. So here you can see that it is so what are the interactions. So now so making so here if you look into it, so the topological module and the functional module and the disease modules, So if we look into it, so now you need to understand that there are two kinds of interaction networks. So this protein metabolite gene genetic interactions, gene regulation, etc., etc., we can do it, but we need to be very much sure. We need to understand that there are two types of the interaction networks that are available, that are present. One is your physical interaction network, and other is the functional association network. So why we need to keep important is to understand these two, because whenever we are trying to cluster each other proteins or particular gene metabolites, protein gene metabolite interactions, so we need to understand that how we are clustering, whether we are clustering on the basis of the physical interaction that they are showing these proteins and the genes and these particular metabolites are showing the very good interactions, or they have co-expression, that when one particular protein expresses or a gene expresses and another protein or gene expresses, or similar those kind of networks. So this is the two things. So for physical interaction network, we have PPI, that is one is your protein-protein interaction network, signaling network, cell signaling and all networks, gene regulatory network. So if you can understand that a protein or can influence after the physical interactions, then is the metabolic networks. And when you have the functional association network, is a co-expression network, and the genetic network that constitutes of your functional association networks. So now in the protein-protein interaction network, so how these databases have been derived, or how one can understand that which proteins are interacting with which proteins. So we need to have a measure or method to understand. So whenever we try to make this kind of network, so what protein is there? So now this protein will interact with how many proteins are the interactors. So what are the methods? So it's better that if you think first that what are the methods through which you can understand that the protein is interacting with different proteins. So one is the tandem affinity purification. So that is, what is that? So you are doing an antibody against that particular protein, pulling it down, and then what you are doing is that you are trying to find out that what all proteins are associated with it. So that will give you the full case of interactors. So another is the is2 hybrid methods. So these are the experimental ways, or the through different experiments what we can understand that how what are the interactors for a particular protein and how many interactors are there. So this will give us generally an overview of our experimental way, but there are also computational ways to understand our protein-protein interactions. So first one thing is the domain interaction. So how we will be understanding that in a in silico way or a computational method to understand that which proteins will be interacting with which particular proteins. So this we can do is to by understanding our the domains of the protein. So if we can identify the domains of our protein, then we can understand through which the interaction of those particular domains, that which one can take it, which ones will be mostly highly interacting with that particular protein. Second is the ortholog interaction. So, and then is the phylogenetic profiling. So phylogenetic profiling will help, because if you have the proteins in a very conserved region or a conserved sequence, so then those proteins on the basis of, you can say, is the topology module or those things. So you can say that it will be mostly being connected with each other. Similarly, is the mRNA or the protein co-expression. So if a protein is showing to have a co-expression with certain protein, then it is highly likely, because if two proteins are getting expressed very much, then it is highly likely that the two proteins it will is getting highly expressed at a particular time. So it's highly likely that the two proteins will get interact with each other. So this is what in the computational method also we can determine except for the experimental. So obviously experimental validation would help us to understand and validate, to understand in a much better way. But the computational methods are also quite effective in understanding that how protein networks can be built upon, or the protein-protein interactions that take place. So either through domain, ortholog, phylogenetic, or mRNA protein co-expression. So now another things that was this is protein DNA interaction network. So protein. So in this we have seen that there are protein, there are gene. In the physical interaction network itself, we have PPI signaling and those gene regulatory networks. So there are interactions between protein and DNA, so that we don't need to re-emphasize. So that we had also discussed with that chromatin immunoprecipitation chip on chip, which chip sake, which if you remember, or you can revisit always the I guess week two or those weeks where we had discussed for the chip sake or the chromatin immunoprecipitation, which showed us the protein DNA interaction networks. But apart from experimental, again, there are means for computational approaches to understand that how we can this protein DNA interaction is occurring. So in the computational approach, like in the for the protein-protein interactions, what was there? The domain interaction or ortholog interaction or the phylogenetic profiling or mRNA protein co-expression. So here in the computational part, what is it is having is the promoter sequence analysis. So why promoter sequence? So where DNA and protein will be binding, so that will have some certain kinds of affinity. Only then it can have certain kinds of interaction. On the basis of that concept, you can do it by having a promoter sequence analysis or reverse engineering from mRNA profiling data. And this is their software where you can try to find out this kind of interactions or the known protein DNA interactions for the different proteins or DNS. So this is the transfact 2.0 transfact. So here you can visit it and try to find out and explore and try to find out that what is the different protein DNA interactions that are there. So if you have any particular protein of interest, you can definitely try it out to find out what kinds of proteins are uh, interacting. So now the thing is that how to get the network. So we are talking about the network. So if you know certain kinds of proteins and the proteins interactions and all those things which are for a particular disease set, so you have done the experiment and have find a certain set of proteins, but how will you find out the networks through which it is related, or to how to get the networks in the co-expression networks. So this is an R package which is WGCNA, which is an R package for weighted correlation network analysis. So correlation. So first of all, we had discussed about the correlation. As if you remember, so correlation gives us that how much a particular variable is related to any other variable. So where you are getting the correlation patterns among those particular variables. So now here what is saying that if you it is doing the same expression level, so according to the expression level you can find the correlation of your genes, that how much it is correlating in for your particular condition. So now you will, what is the rational? So first thing, so how this package works, or what are the algorithm for this package, that which makes you find the weighted correlation for the network analysis for a particular set of genes. So first you will do is that construct a gene co-expression network. So rational is that make use of interaction patterns among genes. So you can see that the interaction patterns among the genes, correlation as a measure of your co-expression. So this is how it is finding. Second is identify modules. So module based pathway based analysis. So you can do it by hierarchical clustering, dynamic tree cut. So all these things take.
दोनों ही join हुए हैं. Hello, Ankit.
It's better if you can type in chat if someone is telling anything.
So whether there is any question, Amardeep?
Is there any question or query? So can you please type in the chat?
Yeah, I think we can go ahead, Ankit.
Okay. So hopefully I'm audible with you. So yeah. Yeah. So where we were telling is that to find out the identify the modules which do the analysis to so hierarchical clustering and the dynamic tree cut, then is to have the relate the modules to external information. So array information, clinical data, SNPs, proteomics, gene information like the ontology, functional enrichment, etc. So it will find you the biologically interesting modules. Then it will have your study module relationship, which will show your biological data reduction systems level view. So eigengene networks and that find the key drivers in your interesting modules. So this is a kind of a package. So now we were understanding that it is going by the module based thing to find your weighted correlation network analysis, so that it will give you the particular interactions networks for any particular kind of a disease. So it will be used for your experimental validation biomarkers and intramodular connectivity and causality testing. So these all things. So technically, these all things we had discussed in some way or the other in the different on different dates of the different weeks of the particular thing. So firstly, so you can understand that how to make the correlation, then to find out the modules which is taking, then to the modules to the external information, clinical data, SNPs, proteomics, gene information, ontology, then to have the module relationships for your biological data reduction, systems level view, and finally key drivers in the interesting modules. So this was for the, so if you ask to get the networks or the co-expression networks for a certain data set or for a certain kinds of gene, so you can definitely try to have one R package like WGCNA for finding your weighted correlation network analysis. Similarly, another what your package is that network serialization and modularization, which is NetSAM, which also gives us the thing like the weighted correlation here. So apart from WGCNA, you can use NetSAM as well. So here, if you go through the documentation of it, so the NetSAM, which is your network serialization modulation package, takes an H list presentation of a weighted or your unweighted network as an input, performs network serialization and modulation analysis, and generates file that can be used as an input for the one-dimension network tool, that is your NetGSalt or other network analysis. So we will be looking at the end of the class today WebGSalt. So similarly, there is a tool called NetGSalt. Apart from that, there is a tool Network Analyst. So these are all the tools that helps us in visualization of the network that has been made. So we will be looking into it, that why network visualization is required. So you can perform the weighted and the unweighted and do all the mathematics and the calculation on the algorithms in the back by understanding all those expression correlations and to find out the disease modules and all those things. So ultimately, what we need to do is that ultimately we need to visualize those tools, visualize those data sets, and visualize those co-expression networks. So this is happening. How is that? First thing is that netsam or the network seriation and the modularization package that takes an so this package will give you it will take in as an input your weighted or unweighted network, and it will perform your network seriation and modulation analysis, and then it will generate your file that can be used as input for visualization like the NetGSalt. So then also can generate your correlation network, example your co-expression or network based then analysis, calculate the associations between the sample features in the modules or identify the associated gene ontology features for the modules. So this is the package, and it also depends on the different modules. You can see seriation, iGraph, WHSC, CNA, which we had discussed, and the uh, weighted gene correlation network analysis. So here you can understand the full form is the weighted gene correlation analysis. So since it provides a certain kind of weightage to all those correlation networks, that's why it is a weighted gene correlation network analysis and bio. So now this is the question. So now these are, you can say that hairballs, like these are the complex networks that generally happen. So this all looks pretty beautiful, but how to infer or how to interpret this kind of hairballs, that which would be looking into it. So before that, if you just see, so this is the whole network. So you can understand this just contains a few proteins or the few reactions and few DNA and few molecules. But whenever a certain kinds of, for example, in a disease condition or a certain kind of model organism, if you check into it for a particular interaction of the, if we say that which particular proteins or which particular biomolecules are being associated with a particular kind of a condition for a particular kind of organism, so you can understand that all these things will be clubbed into one. Then from here it will be interacted with another, then here will be interacted with another, and all these things will be clubbed together, and this will form like this hairball, which I just showed. So if you try to visualize after you perform all in this software your analysis, and it will form the correlations and the correlation analysis and the networks and all those things, then what will happen that when you will try to visualize in the tools that I just mentioned, like the NetGSalt or Network Analyst or in your WebGSalt. So let us try look that if we can look at with some certain kinds of data sets, we also can look and generate this kind of networks or not. Also, there is a very cytoscape. So there you can, if your data set is huge and if you have many genes or things which are perturbed, so you can definitely find out and try to find look at this you will get to look at these hairballs. So and also various manuscripts, if you go and search, you will definitely find that this kind of hairballs have been reported across the globe by different scientists. Now how to infer this globes, these hairballs, so how uh, does how can you, what can you understand from this beautiful networks? So now what is the concept? So whatever we are looking here, it lies behind. So as I have already mentioned in the beginning, that whatever we are doing this kind of network or the networks what is associated with it, it is completely based on our graph theory. So this understanding the graph theory will help us to understand or infer this kind of things, where we can understand which is a node, which is a branch, which is the edge, which is the shortest path, how it is linked. So you can understand that which one, if you can understand which one is the shortest paths among each other, we can try to find out and segregate those things, that okay, so this cluster belongs to these things and will have certain impact, whereas other clusters are very far away from it and will have other impacts and not like this one. So understanding all these graph theories and the concepts of the graph theories is very much important if we try to infer such kind of network theories, where we have a directed graph, omnidirected graph, then weighted graph. So these are networks and modified versions of our graphs, whatever is there. So here you can see with a simple diagram what we can understand. For example, you have a home, you have a grocery store, you have a pond, you have another home, you have a school. So now if we try to map to it, so what will happen? So all the scholars, so from both these homes, student will be so the students of these two particular homes, or the children of these two particular homes, will be in the here in the school. Similarly, in the grocery store, from these two people will go. Then where you will find out. So here you can see that one is your neighbor node one, and this is the complete network. So home, grocery store, school. So generally, what we are living in a kind of a society or anywhere. So whatever, so these are, we are all connected in a kind of a network. So so you all, today's world, everyone is connected through some other means. So all these are networks. So we are all connected through networks. So among these only is the one, it's a biological network. So now if we try to map it, so you can see that the grocery store, then the tree. So this one is a node, this one is a node, this is a junction. So then this is a node, then this is a how it is going to the edges, what is the distance, what will be the shortest path. So all these thing corresponds to a particular kind of network, which is getting reflected in our biological data sets as well, which shows or reflects our biological sense. So now, if what is the concept of nodes edges? So this is a network. So distance is your 2 by a origin, this last distance 5c. So keys here is the edge between nodes, and then shortest path from a to f. So if you want to go to traverse to a to f, you will go through this path, or this path, or this path, or this path. So what is the way? So from going a to f, there are so many possibilities. One can go from this way, one can go from this way, one can go here, 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 then here, one can go here, then here, then here, then here. So now what would be the shortest distance to go from traversing from one node to another node, one edge to another edge? So all these things, distances, will help us in understanding that which ones are closely related to each other, which one has the highest impact, which one has the smallest impact. So all these things are quite linked to each other, if you can understand. So you will be calculating the distance between the two nodes, and then you will be trying to find out that okay, so now if I travel from a to b, what is the distance, similarly from b to d. So in this way you will be finally coming to the conclusion that what will be our shortest path and the edges. And also, if it is, since we have talked about the direction and the directionality of the path of the directed graph, then the another account of the direction needs to be taken into account, that we cannot go in an opposite direction, and all those things. So this is the simplest way of the hairball that we saw just 2 slides back. So this consists of all these kinds of networks accumulated into one place, and that's why it becomes so much scary, and becomes very much difficult to interpret, but can be done if we try to break this all those into much simpler networks. So now the concept is that the network we saw. So for example, here we saw the network, here we saw the network, here we saw this network. So all these are networks. In fact, I'll show. So this is one network. So these are all networks. So now what would be the pattern of the network? So our data sets, when we are talking about a proteomics data set, we are saying that it is normally distributed, or we should have a data set which is Gaussian, which is having a Gaussian distribution pattern. Why Gaussian distribution pattern? So if you say that whatever we generally our proteins expresses in our cells, or the genes expresses in our cells, they tend to be in a Gaussian pattern with having a mean and the standard deviations, which is quite uh, following the trend of the having the same mean, same median, and the same mode. And that's why, and also in the statistical test when we try to perform, so it will have the highest statistical power. So now if you for instance, when we see and when we look at the data types, so after performing analysis for so many types, we are very much sure that we look for that, okay, whether our data distribution is Gaussian or not. In fact, the norm is that whenever if a data analyst is given a particular data, so the first thing that a data analyst will be doing is that trying to find out and do an exploratory data analysis, that is to find out the distribution, the mean, median, mode, that what is the our the data types are there, whether they are ordinal data or nominal data, or what kind of data is it. So uh, this is a normal procedure and the normal process that generally any data analyst should be performing and before going for data analysis. Similarly, it's the case with our proteomics or the transcriptomics general analysis. What we generally follow is to find out the normalization pattern, whether it has been perfectly normalized or not, whether there are any outliers or not, and all those things. So now these networks or these things, so what kind of distribution should this follow? So when I talked about that, these are the four different things. So now, whether, for instance, you take one example. So there is, so if you want to buy one house in a certain locality or a certain network, this kind of network. So from schools, from hospitals, which are your very much crucial aspect in our life, like the schools for children and the college and your hospital for if you become ill. So at what distance would you be taking it to? At what within what distance you should be taking, so that you can follow the shortest distance path, or as well as what would be the distribution pattern would be remaining, or if you in a. So why I told this, so now this will, the distance or the location which will be the prime, or which will be having the shortest distance from school, college, and hospital, that place land, or the price of that land will be become much higher, or the cost will become much higher because it is in the preferred location. So now how to understand what is the preferred location in both the ways, whether the price of that particular area or the land will go high or not. So it will depend upon what is the preferred location of that place. So for this thing, we need to understand that what should be their uh, what should be your distribution of that particular graph, whether you are doing a scaled diagram or a random network or a scale free network. So what is the difference between a random network and a scale free network? So whenever we had if you try to remember, so we look for the t tests and we perform the t test, and we look for parametric and the non parametric test. So now when, so that it depends on the distribution. So similarly in the uh, random network, what it happens is that the number of edges each node has a follows a normal distribution. That means that some network, some edge will have the lowest number of edges and one has will have the highest. Similarly, it will get reduced. So see, the distribution will be same. So this kind of network, when you try to find out the edges and find out the networks in a way that so all will be in a perfectly a kind of a Gaussian distribution with certain have higher number of edges, where the mean, median, mode for those number of edges are same, whereas other edges it won't be same. So these things what you will be finding it out is that you will definitely find that so this type of network is known as your random network. So here you can see that we need to be cautious that often people mistake networks in the world as random networks, but many actually resemble scale-free networks. So before going to the scale-free network, so in this graph there are 12 nodes, 11 edges distributed among the nodes. So node has slightly more edges than others. So node has more edges. Preferential. Okay, so now when we are talking about, so this I am hopeful that it is clear. So in the random network, so all those edges, number of edges will be in a Gaussian distribution, as we generally know that one will have the highest number of edges and others remaining close and within a certain standard deviation, all those edges will lie. But in the scale free network, so again we think that every network is in the kind of a random networks, but it is not so. It is in a scale free manner, and that is obvious, you can understand in the real life practical examples as well. So and as well as in our data sets, for the biological data sets, you will be understanding that it cannot follow the random network. It will have a scale free network. Definitely it will have a scale free network. So this is a scale free network where the number of edges each node has follows a power law. So in the case of a random network, what it is happening, it here the number of edges has follows a normal distribution. But in the scale free network, what it is happening is that it is following a your power law, or it is following uh, so what is power law? So in the power law, it is stating that this graph there are 12 nodes and 12 edges. So also in the there are in fact the theories and the concepts related to that, how many uh, edges are there and how many networks are there related to your particular for a directed graph and undirected graph, how many networks and edges you would be able to receive. So that you can definitely get to it from reading different graphs theory. So here there are 12 nodes and 12 edges, and now but in this case node 8 has many more edges than others because new joiners prefer to connect to them. So that's why you can see that the node B has many new connections. So why I said that in the real life also, in the practical world also, if we think of so as I was giving you the example or analogy of this house and the price land. So definitely if it is in the preferred location, your home or that particular piece of land, then it will be in high demand and everyone will be trying to acquire that pieces of land if it is available. So then definitely a new construction would become will happen at that place. So that will become your center and that will have much more edges than other things. Similarly, if you like someone of has if a person is very much influential or has good personality, then definitely people try to look to connect to that person more than others. So that in that way also it gives you a skill-free network. So similarly, if you try to think, if we all try to think in the line of our proteomics datasets or the transcriptomics or the gene datasets, why it will not follow random network, rather it would follow a skill-free network, is because that genes or the proteins which is at the center, similar to that land and the problem that I said. So if it is in the center and is able to co-regulate a set of proteins or a set of genes in the downstream event, then what will happen is that its effect will be much more. So leading to a network, so leading to a scale-free network. So that's why we would be looking mostly for the scale-free network. So that's why so these things look extremely attractive. But if we dig deep into it, so then we can understand that what is actually meant and how we can understand those concepts. So these are the so this is the concept of the scale-free network and the random network. I'm hopeful that I'm clear. So this is what I was referring to is the biological scale-free network. So biological, if you are asked for it, or if you are if whenever you see network, you can now understand that it is not a random network, rather it is a scale-free network. So why, as I was mentioning, that why your biological data would be, so you can argue, anyone can argue that if the expression levels are in a Gaussian distribution pattern, then definitely their co-expression will be also in their Gaussian pattern only, and that's why we can have a random network, but it's not the case, because the hub having a higher degree or the higher centrality, which can able to influence. So as in the real practical world which we in the see, that the influential people or influential places have your much more attraction, or that the which can attract more edges and the nodes. Similarly, having those have hubs have higher degree, and where you will have the uh, those certainty. So that's why it will tend to take your particular network into the scale-free network. So that's why this is the biological scale-free network. So weighted K-shell. So you can see that this is the range k equal to 1, k equal to 3. So this is in the core. So this is the hub or the centrality. So high degree of centrality or hubness will definitely have a much more impact, because it can impact from here to here longest. For instance, if you think that you are impacting this thing, this particular node, and if there is an impact here, so it is not reflecting to that place. But if it is in the core or the hub of those things, then definitely it will have a huge impact, and it will have on a series of genes and proteins which will have the impact. So this is the weighted K-shell decomposition and the weighted degree of the node I. So that's why, to find out that whether it is in degree, is in the central or is in the node, you need to find out the weighted degree, and then range of 0 node to 1, and then you will find those things. So here what they have mentioned is that biological scale-free network topology represented by nodes, vertices, edges, links in a directed or undirected network. A graphical representation of centrally measures were shown in a hypothetical network. Highly connected nodes, hubs, are shown in black color. So generally we can feel that these are very much easy to understand and to interpret, but sometimes it is not so. We need to dig deep into it, so then only we can get the best understanding out of it. So then you can see that a degree and between a centrality are different types of structural centralities. While eigenvector centrality is an iterative refinement centrality measure determined by the number of neighbors and influence of each neighbor on the given node, represented in purple color. Weighted K-shell decomposition method is an improved K-shell decomposition based on the coordinates of the connected network. So K-shell method or the network is that kind of method which gives you to the understand the coreness, that how much core this your particular gene or the protein lies in that particular network. So left panel represents your hypothetical network with gray nodes, while right panel decomposed hypothetical network into 3 shells. So this is the actual node or the actual network, but it is divided into 3 after we do the K-shell decomposition. So K shell decomposition. So K shell or K nearest neighbor, what we had discussed. So it depends on that how much it can be decomposed into or how much it can be divided into. So the value of K equal to outermost layer with non-significant nodes, green. K equal to 3 represents the innermost layer core of the network with most significant nodes. So as I was mentioning, that the most significant modes, and it is very much similar to what we daily perform, what we daily see in our daily lives, and similar is the case with the biological datasets as well. While K equal to 2 represents the periphery of core with somewhat significant groups. Okay, so now that's why we told. So I'm hopeful that I'm clear and there is no such question. Yeah, so in the network visualization, so that's why we told that we need to have a visualization tool. That's why they have said that after you performed all the analysis and all, so different tools like NetGSalt or these things, here you can see the visualization and can understand the visualization. So now here you can see. So for a network visualization tool, so Cytoscape is a very good and comprehensive visualization tool which you can download in a free of cost, and it gives you a GUI based tool, so you don't require any kind of R or Python knowledge to trying this out. So with the datasets, you can definitely download this free software tool for any given sets of genes or proteins, and then you can try to find out that how the network is built in the cytoscape. Also, I can suggest you that there are many plugins which are available for a cytoscape, and also the protein-protein interactions from string, which provides you the most comprehensive protein-protein interaction database and provides you the associations network. So what you can do is that you can directly link those interaction networks and can visualize in the cytoscape as well. That like what in the WGCNA weighted gene correlation network analysis was given that you can perform the input values in the NetGSort. Similarly, for the string and the protein interactions, you can directly indulge, look into the cytoscape, the visualization. So this is why important. So you see, these are the cores or the hubs, or not the cores or the hubs, rather the clusters like the nucleosome, kinetochore, replication fork. So now you can understand that with the networks and all those things, you can definitely segregate it out, and then in this scale-free network, you can understand that which one has the much more influence and which one is in the center and which one has in the outside, and which will have your much more influence. So now this is one. So as we were discussing the concept like the topology based. So by just visit it once more. The topological module, functional module, and the disease module based prioritization one can do with the genes to form the disease based networks. So here, this manuscript is taking about the prioritization of the candidate disease genes by topological similarity between your disease and the gene profiles, and is topological similarity between disease and protein diffusion profiles. So that's what we were talking about. So you can see that this is a phenotypic available condition and the genes responsible to it. So this which will can calculate in a certain kinds of equation the association between the phenotype and the particular gene, and then you can perform the in the mathematically. So next is the, so this I have given another example which through which you can perform the candidate gene prioritization by network analysis of your differential expression using machine learning approaches. So why we I had put into it or kept this manuscript, because we have at least discussed to some extent different machine learning approaches. Like in the previous week also we had discussed like certain concepts of the classification. Similarly, we have done some unsupervised clustering algorithm, some concepts. So then you can understand that how, what are the things that we can take it forward or do it to understand this kind of gene prioritization. So because this becomes very much important for us to calculate the network and construct the networks. So now another thing. So now till now what we were discussing is that we are getting, for example, getting the different sources or through different experiments. We know that physical interactions are happening, or through computational methods. So like your is to hybrid system, or for your wet lab techniques, or you have your tandem affinity purification, etc., etc. What we were knowing that these are the physical interactions, protein-protein interactions, and those genetic regulatory interactions, as well as we are performing domain prediction, ortholog prediction, etc., etc. We are trying to find out the what are the possible interactions. But if we don't know for a novel disease genes, so for a novel disease genes, what would be the best way to have? Because with the known disease associations, which I have already shown, the human disease network or the human metabolic disease network, so one is aware of the different diseases or the genes of the different, like the OMI and the literature is available, which one can easily look for and check for those kinds of diseases. But if we say that the novel disease genes, which disease we don't know, or the network of which we don't know, for that, if we need to look at the genes having their maximum association, so how to move ahead with it. So that's why I have given this kind of this manuscript, which says that how we can do for the differential expression using machine learning approaches. So that's why we had discussed to some extent the different machine learning techniques. So now here what we can see is that look that how they have performed using they have used different machine learning approaches for the network analysis. So here what it is there is that it discovering the novel disease genes is still challenging for diseases for which no prior knowledge, such as known disease genes. So you can understand that no disease genes or disease related pathways is available. Performing genetic studies frequently results in large list of candidate genes, of which only few can be followed up to further investigation. So this is a very real statement or real problem. You can understand that if the all the genes or all those things which are coming, so all those things cannot be taken forward for your analysis or further investigations in a different cohort of samples or different things. So definitely we need to choose a list of candidate genes through which only few can be followed up for your effective further investigations. So they have recently developed a computational method for constitutional genetic disorders, identifies the most promising candidate genes by replacing prior knowledge by experimental data of differential gene expression between affected and healthy individuals. So this the computational method that they have developed. So this is just replacing the knowledge of the prior knowledge of the experimental data of different gene expression between the affected and the healthy individuals. So what they are saying is that to improve the performance of our prioritization strategy, we have extended our previous work by applying different machine learning approaches that identify promising candidate genes by determining whether a gene is surrounded by highly differential expressed genes in a functional association or protein-protein interaction network. So just think that what concepts they have brought into place is that if they have tried to identify promising candidate genes by determining whether a gene is surrounded by your highly differential expressed genes in a functional association. So if it is in a, we have already discussed that the physical interactions and the functional association, because of physical interactions we have discussed, like the protein-protein interactions, protein-gene interactions, and all genetic regulations and all. So similarly, if it is a gene is surrounded by highly differentially expressed genes in a functional association, or the protein-protein interaction networks. So there, what we are doing is that we are getting that uh, uh, what you say, that the protein-protein interactions what we are actually getting. But so so now if the your candidate genes is present, so if your this thing is present in your candidate gene is highly surrounded by differentially expressed genes, and or it is present within a protein interaction network, so that will give you the priority. So now three strategies scoring disease candidate genes relying on network based machine learning approaches. So what machine learning approaches we have seen to classify or to identify or to select the features for your set of candidate biomarkers, or the markers that are really showing the differential expression. So relying on network-based machine learning approaches, such as one is kernel ridge regression, heat kernel, and Arnoldi kernel approximation. So these are the things. So I am hopeful that by going through the previous week's class, so you can understand the problem between the ridge and the regularization, and the lasso regularization and those ridge regression. So I am hopeful that you are clear. So here what they have done is that the gradient descent and the regularization, what is the major difference. So I am hopeful that you all are clear. So now this terminology will not be so much unfamiliar to all of you. So in the kernel ridge regression, which is a kind of a ridge regression, but having a weightage or kernel, or then heat kernel and Arnoldi kernel uh, approximation. So they have used these three techniques. So now for comparison purposes, a local measure based on the expression of your direct neighbors is also computed, and they have these strategies on 40 publicly available knockout experiments in mice against results obtained using a standard procedure in genetics candidate genes which only on the differential expression level. That is your simple expression ranking, that on the basis of your differentially expressed you will perform those rankings. So their results have shown that our four strategies could outperform the standard procedure and the heat kernel diffusion ranking, leading to an average ranking of a position of 8 out of 100 genes, an AUC value of 92.3% and error reduction of 52.8%, which ranked the knockout gene in average at position 17 with an AUC value of 83.7%. So they have identified promising candidate genes using network-based machine learning approaches, even if no knowledge is available about the disease or phenotype. So they could try to, with the publicly available knowledge basis and the diseases, so they could after uh, finalizing machine learning model approach, you can see that the machine learning model approaches are required or can be used in this kind of network constructions as well, and it's very much good or very much beneficial when there is no prior knowledge about a disease or the genes. So this was all about the network. So before we move on to the so I am hopeful that there's no questions. Before that, I would just like to just give a revision. So we have So the networks are very much important for us to understand mm, that we are uh, how we are going ahead and how we are getting what are the different sets of what you say that the how different sets of things are going on. So to have a full look of the knowledge, so that's why the networks, because to understand at the omics level, we are very much capable of understanding the all the things that are happening in unison. So now, so all those things, complex networks of the complex hairballs that we look upon on daily basis. So what is the need or how we can understand it is the basis of the graph theories. So now we have also understood that the concept of the shortest path and the degrees and the nodes and the unidirected graph and the bidirected graph, undirected graph, all these things will lead to understand that how many edges are there, nodes are there. Next is that what is the distribution of the networks that we generally see in the kinds of our data analysis, but we don't try to overlook into this kind of network. So we can see that it doesn't follow a random network, and it follows a scale-free network, which is quite practical in our real lives as well. So all those networks, what we see and what we follow, are generally not kind of those random networks which follows our normal distribution with the edges and the nodes. Rather, all are scaled in a positioned in a scale-free network. Then we could understand that the degree and the coordinates of the, if we can find out the prioritization in the kind of code, if we go to the code of the particular things, so then definitely the influence and all those things will be high. Then finally what we could look into it, the different approaches which can be done. One was the topology based and the protein diffusion based strategies, which taking into the mathematical equation. But another interesting thing, what so there are many things that you can always explore and can try to look into it, but I gave one showed one example which was very interesting for the novel disease in which we don't have a prior knowledge or prior literature, then how to move ahead with it. So they have used simplest machine learning technologies to some extent, which we had discussed in the previous lecture. So which is not so much unfamiliar with you all. So they have used different algorithms and tried to use it in the publicly available data sets, as well as what they have done. They have done on the simplest your simplest ranking system, that is on the expression level for a set of a genes. So how that you are getting. So we are clear till now of the network systems and the network things. Before we see some real aspect or the some kind of aspect in the sense of in the network topology analysis in WebGSalt or network analysis. Let us try to find it out till the end. But before that, another important thing in the proteogenomics is that we have talked about that SNVs, single nucleotide variants, or the SNP, single nucleotide polymorphisms, and their effect and their conversion into single amino acid variants. The SNVs to single nucleotide to amino acid variants, and whether that variant that happens at the amino acid level is synonymous mutation or non-synonymous mutation. What are those things? That becomes very much crucial. Now there's an important to understand that what is the motives. If in the, for example, in the kinases, so if we say, so you all can understand the importance of the kinases. So they play a major hub or role in the phosphorylation. So kinases plays does a phosphorylation, and phosphorylation of a particular protein or dephosphorylation or not letting you to the phosphorylation can lead to a cascade of chain mechanisms being disrupted in your biological system. So now, if it happens that the single nucleotide variants or the single amino acid variants, if it occurs in a certain kinds of thing that the kinases gets altered, then what is will be the effect, or how we can find out all those effects. So this is a thing which was given, that the biological sequence motif discovered using motif X, so motif X, to find out that what is the effect of those SNPs or SNVs onto the kinases. So before going to that, let us refresh certain parts of what is a motif and a domain. So just from the basic bioinformatics, if we just brush up, because if some of you is unfamiliar with the terms of motifs and domains, so what exactly it means. So motifs and domains is an important way to characterize unknown protein functions. So motifs, so why suddenly motifs? So that's why let us see that what is actually a motif and how, what is the definition of it. So to characterize unknown protein functions, because only a newly obtained protein sequence often lacks significant similarity with database sequences of known functions over their entire length, which makes your functional assignment difficult. So what is that? It is a consensus sequence patterns. So motif is your short conserved sequence pattern, which is associated with distinct functions of a protein or DNA. So what happens is that there is a conserved regions sequence pattern for a particular function of a protein or DNA. So the protein having a particular function. So for instance, why the concept of motif came for understanding those in the kinases. So the kinases, it's believed to have, since they are playing a similar kind of role, so they will have a certain kind of conserved regions. So since they will have a certain kind of conserved sequence, but a short conserved sequence pattern with a distinct function of the like the kinases doing the phosphorylations. So this will give you the motifs. So understanding or finding that motifs will lead us to means it will help to understand that how it is faring. So this is one thing. Second thing is that another difference is the domain. So we have also today said that we will be the computational methods are generally used apart from the experimental data. Computational data uses the domain for the finding of your protein-protein interaction. So what is that domain? So that's why I bring it to here as well. So if you have any doubt between your motif and your domain, so this is also a conserved sequence pattern, which is an independent structural and functional unit. So the major difference between your motif and domain is that, so if you compare motif on one hand and the domain on the other hand, so both are conserved sequence patterns, but the major difference is that domain is itself a complete structural functional unity. The length of the domain is such that it is able to produce, it is able to function and do it individually, but motif is not. It's just a short sequence concept pattern, mostly a kind of a signature for those particular kinds of proteins or the DNAs having those particular functions. So example of motif is, you know that so it is zinc finger motif, which consists of about your 20 amino acids long, associated with a distinct structural site and associated with a particular function, like the zinc finger motif, which gives you around about 20 amino acids long, and it's associated with a structural site and associated with particular protein function. And domain is definitely which one is a longer. So the way we have spoken about domain, so the domain by definition is longer than your motifs, consisting of about 100 residues in your average. Why 100 residues? So you can understand that are independent units, structural and functional independent units, and evolutionary more conserved than other regions of a protein. So that's why now you can understand. So if it was unclear at that time that why domain studying and the domain interactions would help us in identifying those proteins. So if we can analyze the conserved sequence, so it is more conserved part. So if the two proteins have the same domains or a similar kind of domain, then definitely they would be having a certain kinds of interactions or would be closely associated with each other, because these are domains are the evolutionary most conserved regions than the other regions of the protein. So that's why, and this is the major difference between your motif and the domain. So domains are long, motifs are short, and the domains are independent structure and functional unit. So now, what are the how we identify our domains or motifs? So what is the concept behind it? How we can try to find out the motifs and domain. So identification of our motifs and the domains relies on multiple sequence alignment as well as the construction of your profiles and the hidden Markov models. So why we are discussing it? Because we want to see that the kinesis motifs we try to understand and try to find out the effect of the SNPs or the on the kinases, which will play a huge role in further steps. So motifs and domains are first constructed from multiple alignment of related sequences. So you can understand that if you try to identify certain conserved regions of those sequences, then you need to first align those related sequences. Then after that, based on those multiple sequence alignment, you will be taking the commonly conserved regions and identify those commonly conserved regions. Then is the consensus sequence information of motifs and the domains stored in a database of the presence of similar sequence patterns from your unknown sequences. So you can understand that the functional features in a query sequence. So if you are interested more into it, you can definitely go and have a look at the motifs and the domains in a using. But the major take home here is that the motifs are the short conserved sequence of the DNA regions, DNA or protein regions, but they are not structurally available for you to be independent. But domains are long and they're structurally, and it's the most conserved regions of any particular protein. So that's why also we are looking at the PPI through computational methods by identifying the domains. So motifs or domains is that it will have your consensus sequences as regular expression, whereas domains it has by constructing statistical models such as profiles or hidden Markov models. These are the different statistical models that you can take forward while understanding the domains. So this is just the consensus information of the motifs and domains from your reduce the multiple sequence alignment and the regular expression. So here I have given you the protein phosphorylation motif or the motif places where can be phosphorylated are like your ST, X or RK statistical models such as profile or HMM. So this was motif X, that so biological sequence motif discovery using our motif X. So let us see that what they suggest. So you can see this is the phosphorylation data sets. So this has been given. So this is an experimental data set. So now if you get a phosphorylation data set, that is your experimental data set. Now what you are doing is a background data set. Okay. So this is the background data set. So now what you are doing is that step one is your recursive motif building. So that's why I gave you all the definition of the motifs and the definition of the domain. So then how the regular expression and the multiple sequence alignment is converted into regular expression. Sorry, regular expression matching, so that you can understand that, then only you can understand that how this algorithm works or how they suggested to find out those effect of the kinases. So step one is your recursive motif building, where motif building, where you have the data set frequency matrix, background probability matrix, and then you calculate your binomial probabilities. Then you will do the most significant residue position or pairs. So then removal of sequences containing motif discovered from the data set and background sequence list. Then step 2 is the set reduction. You will continuously reduce the steps. The algorithm is complete when the binomial probability matrix reveals no significant residue. When there's no significant residue left, then it will automatically get exited and here it will be finalized. So this is a kind of a thing through which you can try to find out that how the phosphorylation will be affecting the kinases for the particular motifs. I am hopeful that you get some idea regarding those things. Next is that I have given this example of the that where you can find out the endogenous kinase substrates by proximity labeling combined with kinase perturbations and phosphorylation motifs. So if you want to explore much more about it and want to learn regarding it, so you can definitely go and explore this paper to find out that how they have tried to find out the kinase substrates and the kinase perturbations and the phosphorylation motif. So that's why I had also given you the protein phosphorylation motif, that how it looks like in the regular expression pattern, which what is the effect on this. So generally what we are doing is that we are looking at this motif region for this particular protein sequences. So what when we are doing any kind of perturbation on the kinases, so what is the changes that we are looking at those particular motifs level. So that's what we are looking upon, or what we are interested upon. So now next thing is that, so this is another tool which is known as your MIMP, or predicting the impact of mutations on kinase substrate phosphorylation. Okay, so this is in this paper or in this tool, what we are doing is that to predict the impact of mutations on kinase substrate phosphorylation, that what we have just discussed, that what we are interested upon. So here what it is given, that the protein phosphorylation is important in your cellular pathways and altered in disease. So the authors have developed MIMP, which is online available, which you can definitely go out and try and look into it. So this is the link which is there. So again, you can see that the machine learning method to predict the impact of missense single nucleotide variants, what we just spoke about, that machine learning method to predict the impact of your missense single nucleotide variants on kinase substrate interactions. So on the kinase and the substrate. So the what will be the substrate? That the phosphorylation motif. What we have just shown you, that how it looks into the regular expression levels, which is derived after your multiple search. What is that? Multiple sequence alignment. So MIMP analyzes kinase sequence specificities and predict whether SNVs disrupt existing phosphorylation sites or create new sites. So MIMP or MIMP, whatever you say, this catalyzes, analyzes your kinase sequence specificities and predicts whether SNVs disrupt existing phosphorylation sites or it creates your new sites. These help discover mutations that modify protein function by altering kinase networks and provides insights into disease biology and therapy development. So this helps you to discover mutations that modify protein function by altering kinase networks and provides insight into disease biology and therapy development. So these signaling networks mediate complex cellular logic, so you can understand, often by modular protein domains and short linear motifs. So already we had discussed and I have given you the definitions of the domains and motifs. So you can see that the modular protein domains, so domains is always your long and which is structural and functionally available, and the short linear motifs, so which zinc finger motifs are the short regions of the conserved sequences, but the most conserved regions remain your domain. So the impact of the genome variation on molecular interaction networks, difficult, however, interaction involving short linear motifs are well known enough to tackle. Okay, so you can see that the impact of genome variation on molecular interaction difficult is difficult to predict on the whole molecular interactions. So why I am just giving it so that you can uh, get the idea and what is the implication of this kind of tools and how much effective is to understand specifically in the cases of the disease biology and the therapy development. So involving short linear motifs are well known enough to tackle. A prominent example is protein phosphorylation or post-translation modification of threonine or tyrosine residues. So you can go through it. So these motifs are inferred from networks of experimentally confirmed phosphorylated sites and the associated kinases. So phosphosites are evolve variants in phosphosites and other protein sites, but it does not provide you methods to automatically predict the impact of mutations on the kinase binding. That's why we need to assess those. So leverage rapidly increasing genomic and phosphoproteomic data to interpret variation in the signaling network. So you know that the phosphoproteomics data is also getting evolved with very much robust instrumentation methods. So these kind of methods and the variations and trying to find out will be increasing to a great extent. So this method which called is the why what is the meaning of the meme is that mutation impact on phosphorylation to predict the function of SNVs in phosphosites. So this is on the method based on Bayesian statistics. This is again a machine learning based tool that is dependent that is here in the Bayesian statistics. So this is a kinase associated from public databases, constructed position weight matrix models of amino acid specificities of kinases. So similarly, like your multiple sequence alignment, so this at which position, all those things are there. We modeled 124 high confidence kinase with at least 10 known phosphosides, including 99 serine kinase, 25 to in kinase, 58 kinase for families, etc., etc., and all those things. So this you can go to this paper of this nature biotechnology, and you can go and find out all the details. Also, you can definitely try to find out from the web version that what how you can look for it. So we will be looking at another tool. So this is what workflow is generally there for the meme for the mutation impact for the kinases. So you have the phosphosite collection. So you will have the positive kinase phosphosites, random negative phosphosites. So why you are giving negative phosphosites? So that your model understood and doesn't get your overfit and all those things. So you will construct your model identifying PSNVs, wild type phosphosites, mutant phosphosites, predict your rewiring events, the loss and the gain, interpretation, and then experimental validation, and then you will find the what are those actual places where your it's getting affected. So you can read this paper in a much more detailed way, which will give you those answers, and also you can try it out by going to those particular place. So this is another paper. So I have again put into it as a resource, which if you are interested into it, obviously you can look into it and can look it forward. So this is phospho-peak SNP, that is quantifying the effect of amino acid variants on protein phosphorylation. So the GWS also, though this is also one kind of paper. So this perturbations or this, since we are looking at the proteogenomics approach, so why these two technologies becomes very much crucial and important for us to understand. So it also becomes understand also in the major level like the kinases, what is the effect of SNPs and the single amino acid variants. Now this is another tool which is the active driver DB, find your mutation of the genes of interest. So now we will let us look into it in a kind of a, so let us So this is a web-based tool. Yeah, so this is which you all can also search. So you can see that here given that it is you are viewing the active version, updated version. So find, so you can see that proteins, mutations, pathways, PTM sites. So what is your active driver DB? So it is a human proteogenomics database. So these are all resources which I am just giving you an overview, which you can definitely go back and look into it in much more detailed manner. So these databases uses informations about PTMs in proteins to annotate and interpret genetic variation, genes and cancer driver genes. So this is a classical example of what we can do it through proteogenomics approaches to what we can look at the cause and the effect simultaneously. So all datasets in active driver DB is collected from public resources and based on experimental data. From some examples, dementia, cystic fibrosis mutations, cancer mutations, interactively downloaded in bulk. Okay, so this you can find out. So data, proteins, kinases, mutations. So you can see that what we have already spoken about in one of the weeks, that we have took 1,000 genomes, PCA, So these came recent. Then the TCGA, clean war, mutations, pathways, all these things. So they also provide you the tutorial. Okay, so there you can go through it. So you can go for the protein search. So you can just quick run examples. So example, you can see the sequence and the network. So was used to visualize mutations affecting PTM sites in human proteins. So wherever the PTM sites are affected in human proteins. So in fact, what we can do is that go for search mutations. So here you can select a VCF file as well. So I am hopeful that you all know what is a visual VCF file. Yeah. So network rewiring, motif loss, proximal, distal, impact. Direct is PTM, true, all are true. At what reference, what alter, what position, what is the link. Okay, so the what cancer type, TCGA, CAWG, pancreatic cancer, tumor, hereditary cancer. So all these things you have got that from where the experimental data is coming. Okay, so this you can go ahead and look at active driver database, which are the things in the PTMs. So you can load an example as well. If you are interested in certain proteins or genes which you are aware of, you can definitely do that and try to find it out. So this is a excellent tool through which you can find it out. Okay, so now let us move into it. So this is the publication through which you can find it out. So now the different what we have talked about, the different pathways. So now, so firstly from today, in today's week, what we had discussed was, firstly we have discussed about the networks. So in detail about what is a network, what is the nodes, what are the edges, what are the graphs, what is the shortest distance, how to interpret and hairball. So what are the biological networks, protein-protein interactions. Then you our what we call it as our this functional annotation, functional networks. What are they associated with it? And then is it a random network or a scale-free network, biological networks, or what kind of networks? And all those things that we have discussed. Then we have looked upon to identify that an effect of the mutations, on a particular protein phosphorylation sites. That's why we have undergone the different motifs, then the domains, and then have tried to find out different tools that are available. One is your MIMP, one is your active driver DB, so which I just showed that you can get all those things networks. Now the third most important, after you got the network, the protein-protein interactions and the protein DNA interactions and the metabolic networks, all those are interconnected. Now, what are the pathways? That's a fatty acid pathway, it's a amino acid pathway. What is the exact mechanism or the effect that is being taken place? So, whether you will get inflammation will happen, or what kind of fatty acids will be altered, lipids will be altered, bile acids will be altered. So because all these alterations or the metabolite level after the genomic changes and at the proteome level changes, when it goes at the level changes of those particular metabolites at that functional level, so then it has also an adverse effect on the localized regions or the localized, like the inflammation or the particular liver damage or those kind of things for the human, or similarly for other organisms as well, or for different plants and microorganisms at the different, they are producing capacity, they are this all these things can have a huge impact, the local things. So these are different databases of the sources that you can definitely go through it. So like the MSigDB or the molecular signature databases. So I am hopeful that correct me if I'm wrong, that I had shown you the MSigDB database and also the gene set enrichment analysis. And in the previous week, we have discussed in detail the regarding the definitions of our overrepresentation analysis and gene set enrichment analysis. Whereas in the overrepresentation analysis, a certain set of upregulated or downregulated genes, if it is present in that particular pathways or how much they are present, so that defines our over representation, or if it is over represented or not represented a particular pathway. But in the gene set enrichment analysis is the sets of the genes that means actually getting deviated on particular pathways. Apart from the statistical significance, it is taken forward, and the number of edges and the nodes which are related in your particular study for a change in a particular disease on the basis of their expression levels is taken forward, which is known as your gene set enrichment analysis. So now this, I am hopeful that I have shown. If you all don't recall, just go back and check in the videos that we regularly upload in YouTube. So you can go and find out that how those this GSA pathway looks like that. So similarly, in the MSigDB, this molecular signature databases contains many pathways and the genes that are associated, the genes that are associated with this particular pathways. Similarly, is the cake, which gives us the Kaito encyclopedia of the genes and the genomes. Then the wiki pathways. So and then the reactome. So these all, so wiki pathways is the kind of Wikipedia of the pathways that are there, which gets perturbed. Similarly, the reactome, we can find out different pathway browsers and the analysis tools. So now today, so these are all the sources which you can go and explore on your free will in as much as possible. Yeah, definitely. If we get time next week, we can show the reactome and the different pathways. But today, previous week in the WebG salt only, what we had done is that we have taken the data and performed the over representation analysis. So today we have talked a lot about networks and those things. So let us try to look into the network topology, how we can get. So what I will do is that, so active driver you can Okay, so here you can see that there are three options. So definitely what you can do is that you can go and find out network topology based analysis. So functional database is network. So I can give PPI. So now you can understand it's a PPI is a protein-protein interactions and the TCG, RNA-seq, all those things are given. So if I get uniprot, swissprot, so what I will do is that, so I will take again the dataset. So here the concept is that we should go for the statistical analysis, missing value imputation, normalization, and then try to find out the differentially expressed regions of the proteins and the genes, and then go for that analysis. But here let us take some 2025 random for this purpose to show you the functionality. So here you can see that the network retrieval and prioritization. So that's why we had discussed all those things. So now it will show you the network analysis. So if you go, so meanwhile, what I will show is that another tool that you can use is the network analyst. Okay, so here you can multiple gene expression table, raw RNA-seq data you can give. So specify organism, so if you give humans, ID type, uniprot protein ID, and then you provide here. So you can see generic PPI, tissue specific PPI, host parasite or microbiome based PPI. So in this is the network analyst. TF gene signaling networks, protein drugs. So let us look into the generic PPI only. So again, it is giving you the database. So all these are resources. So if you go and search each all of this, so all will have resources. You can see Roland Interactive, Hurai, and all those publications. So let us go for string. So what is the confidence score cutoff? So we will keep at highest. Require experimental evidence. Okay. So this you can change accordingly, whatever required. And then you can proceed. Yeah, so you can view the topology. So if you go for the first run. Yeah, so you can see that the network and the hairballs that we were discussing, you can see that it has been formed. So whatever data sets you were getting, so how much is the degree? Now you can understand that how we will understand that which one is at core or how it has calculated it. So which one will be getting in your core as well as in the degreeness, centrality. So that's why all these things is coming into the place. So then you can do query for the upregulated nodes, downregulated nodes, Kate, reactome, gene ontology on the different basis of it. So you can see that now which one is at level edges and all those things, and you can explore. So the all the hairballs that you are looking now, you can also make it and can use it in your research, and not only use it, but also what you can do is that you can interpret it in a much better way. So the explorer. So you can specify your view, you can proceed, then node you can specify, age you can specify, the color and all those things. So similarly here also we will get, so the results or the topology file like that, which you can definitely take it forward. Sorry. So this is two things. One was the network analyst and web result, and I'm not hopeful that NetGIS solved which they said. Okay, so this is your NetGIS solved, that what we had discussed. Flexible data integration. So here also they are giving you the track for visualization and the visualization tools. So what you like to do? Analyze your own data. Human. So they are providing only for human and mouse. So you can use accordingly and then finally provide. So you can see that they also take human this transcriptomics data or the microarray data for those things, and then you can perform. So that that is net SAM, that serialization and modularization. So if they provide you the data, you can directly take into net results. So there, I think it's showing certain kinds of things, but definitely if you provide this gene names, so if we say no genes are mapped, so definitely we have to give universal, but so if you just submit, yeah, so if you just submit, it will definitely sometimes the web-based tools have some issues related to the server, and sometimes it throws up error, but it will definitely give you the things. So is there any question as of now?